Hello, everyone, and welcome to today's live broadcast, Next Generation Sequencing to Understand the Function of MicroRNA in Human IPSC-Cardiomyocyte Disease Models, presented by Ulrich Broeckel, Professor of Pediatrics, Medicine and Physiology, and Amy Turner, Project Leader, MicroRNA Technologies, Research Associate 1, Department of Pediatrics, Medical College of Wisconsin. I am Sarvi Greneman of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, visit www.qiagen.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located on the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Ulrich Broeckel and Ms. Amy Turner. I will turn the presentation over to them.
Great. Thank you very much for the nice introduction, and I would also welcome everyone to our presentation today. So we're very excited to talk about the research in our lab and the work we are conducting using human iPS-derived induced pluripotent stem cell-derived cardiomyocytes as a disease model. And let me just start with a legal disclaimer here, which we need to post related to some of the products and technologies we're using from our sponsor today. In this presentation here, just a brief outline. The topics of what we're covering today, we will talk a little bit about disease modeling and the role of disease modeling in precision medicine, the use of human-induced pluripotent stem cell-derived cells, in particular cardiomyocytes. We'll talk about a study cohort which we have been working with at the National Heart, Lung, and Blood Hypergen cohort, where we have been focusing and studying the genetics and genomics and precision medicine for related to left ventricular hypertrophy. We will talk about our approaches to use RNA sequencing expression analysis for RNA and microRNAs in relation to our disease phenotypes. We'll talk and introduce our overall experimental study design, how we measure disease in our stem cell-derived cardiomyocyte model, and we'll talk about some of the methodology we use for, particularly for microRNA sample processing and sequencing, and discuss with you our analysis approach and the experimental results, and provide some conclusions for this. And so let me just start. Precision medicine is now really gained substantial momentum in terms of how we would like to understand and really describe disease better. And it really started out, obviously, as many of you know, and for many years that genetics and genomics and understanding the genetic basis of disease was really the main focus. But I think as the field matures and we really learn more about the impact of genome variation on disease phenotypes, we would clearly recognize that just looking at DNA sequence itself is really not sufficient to really describe the complex disease processes which differentiate patients and to categorize disease better. And so clearly the next steps in precision medicine to really to advance the field is really go beyond just genome sequence and really combine the genomic information also with functional analysis and next generation sequencing, particularly expression analysis, where we look at RNA and microRNAs and then really extend this to proteins. And if we really look at the overall circle where we start with a patient, as I said, we're looking at DNA, we're looking at DNA sequence, Then the next cycle, we'll look at gene expression, microRNAs, we'll look at proteins, transcriptomics as an analysis approach, and proteomics. But we'll move on to actually look at cells and these phenotypes in cells and tissue themselves, and then extend this and combine this again, looking at the more as an organ system and multi-organ systems, and then really bring this together and describe the disease phenotype in our patients much more comprehensively. So it's really combining all those things together. And in precision medicine, and as you can see, if we compare the more epidemiological approach, where we're looking at just general risk factors, where we're looking maybe just have one drug to treat, or we're looking at one disease by themselves, we basically categorize patients just as either have the disease or not have the disease. And that often epidemiology population-based approach use this type of concept. And we really look at the disease more as a very general disease model. But if we're moving towards precision medicine, I think what we need to include in our analysis and model and really take into account is that every patient has a specific environment. We need to be very specific, and also in the way how we treat a patient, for example, with drugs. And ultimately, the underlying concept here is that every patient has, in many ways, a very specific disease presentation. And that really then leads to this concept, what precision medicine really stands for, that we're looking for a more individualized and targeted disease treatment approach. And if we want to study a disease, that obviously, this concept then leads to kind of the requirement that we really need to be able to model and develop disease models which are unique to a specific patient. And so the foundation, how we pursue this in our laboratory here is using the technology of induced pluripotent stem cells. And just to review here this very briefly, you start with a patient, you collect somatic cells, in most cases now a blood sample. You culture these cells, you add 4 distinct pluripotency factors, and these cells in culture then actually regress and become, again, pluripotent cells, induced pluripotent stem cells. Now you can take these cells, differentiate them out in any type of cell type you're interested in. Now we're particularly interested in cardiomyocytes, so these cells get differentiated in cardiomyocytes. The advantage of this is that the derived cells and the iPS cells are really as unique as the individuals where you've taken this from. We basically carry the genome in the model, and particularly if you're looking at, for example, a genetic disease or a disease which is determined by genes, by and large, these cells actually do recapitulate phenotypes related to a patient's genetic risk. And if you then do this not only for one cell line, but for multiple cell lines, and use this as a disease model, I mean, the advantage here is if you start with your patient, you make these iPS cell lines, you differentiate those in your target cells, establish a disease model. What you can do here is then you can do very comprehensive phenotyping. You look at the disease phenotype. These cells are obviously also used for drug screening, for biomarker development. And in many ways, you can say I'm diagnosing these cells in the tissue culture, and as you treat them with drugs and look at drug response, you can also look at the response of your treatment. Now, the advantage is if you characterize your patients well enough, you also have this data available. You have biomarkers, you have very comprehensive phenotyping, and the really major strength we see in this approach is that you can actually validate what you measure in your cell culture with what you measure and the phenotypes and disease presentation, what you see in your patients. And compared to, for example, animal models, this clear correlation and comparison, validation of your biomarkers, I think is one of the major strengths of this platform. Now let me introduce to you the disease we're really focusing on. And for many years now, we've been studying the genetics and risk factors of left ventricular hypertrophy. It's defined by an increase of the weight of your left ventricle. And as you can see on the diagram here on the right side, the major causes, underlying risk factors are high blood pressure. We see this also in patients with diabetes, kidney disease, and age and gender, in addition to race, are well-established risk factors. And left ventricular hypertrophy is now well established as a risk factor, for example, in patients with hypertension, with high blood pressure, as a risk for hypertensive heart disease, which ultimately leads to heart failure. In the next slide here, you see already kind of a summary of the expression changes in genes and pathways which have been linked to cardiac hypertrophy. And as you can see here, this is quite a comprehensive and complex picture, including many different disease and networks really contributing to our disease phenotype. And in the following slide here, just also a summary what we know so far about the microRNAs, which have been linked to cardiac development, play a role in cardiac development, cardiac regeneration, and then on the left side, as you can see, a cardiac hypertrophy. So we already have kind of a rough framework of genes and microRNAs and mRNAs, which play a role in disease process in more of a general disease model. To now dive a little bit deeper, to look actually again into individual variations in these overall models and dissect this a little bit further. As I told you before, we're using iPS-derived cells, and we're working with the Hypergen study. And part of an NHLBI funded grant, we actually generated 250 iPS cell lines from a subset of individuals which have been recruited for the Hypergen study. The Hypergen study itself is a biracial cohort. We have African American and Caucasian participants. We have very comprehensive phenotyping. It's a family-based study. And we already did genome-wide association studies. We also have whole exome sequence data available. So it really builds the foundation for our genetic analysis. And so we selected a subgroup of 250 individuals and generated their iPS cell lines. And that really builds the foundation for our analysis and for the experiments we're discussing today. And the next step is obviously we have to model also our disease phenotype. And what we did over the years is we actually established a high throughput phenotyping platform and pipeline to characterize these iPS-derived cardiomyocytes. So we culture these cells in 96-well format. We induce hypertrophy. This is kind of an environmental challenge if you want to look at this way. We induce them with well-established inducers of hypertrophy like endothelin, phenylephrine, or isoproterenol. We culture these cells, and at the end, really our main phenotypic readout at the end of about a 4 to 6 week protocol is expression profiling, but then also following downstream potential protein analysis. And that really allows us to look at pathways, to look at networks, and understand the variation between the different individuals and different cell lines which represent individuals in much greater detail. So the next slide, one of the questions could be is like, do these cells actually develop a hypertrophic phenotype, these cardiomyocytes? And one way to look at this is, for example, measure the cell surface area. What you see here on the left side here, if we stimulate these cells with different stimulants compared to control, they clearly increase in cell size. We can also look at CFOS, which is a early immediate gene. And there as well, you can see that the cell lines stimulated with our well-established stimulants, inducers of hypertrophy, these cells actually do increase and change their fluorescence for CFOS. So we also did some analysis where we looked at the expression changes related to differences in microRNA. On the left side here, in red, you see a number of microRNAs which are differentially expressed in our model of cardiac hypertrophy in a cell line. So what are really the next steps now as we're looking at microRNAs? Obviously, we can look at how, and in these different cell lines now, where we try to understand the variation better between different cell lines and the differences between different individuals. And obviously, as we try to understand the variation in the RNA expression in different cell lines for multiple individuals, and we can do this in patients with and without, or in individuals with and without hypertrophy. Ask the question, what is the inter-individual differences? What are the phenotypic differences? We can look at, as we're analyzing mRNAs and microRNAs, we can look at intracellular interactions and dependencies between mRNA and microRNA, and what are the genes which are affected by that between the different individuals, what pathways are affected, and then we can also look at predicted targets, for example, of extracellular microRNAs, and we'll explain a little bit more our analysis approach to microRNAs as they get excreted from, for example, these cardiomyocytes as they get excreted in exosomes. And so you can ask what genes are affected by that as well, and what pathways are affected. And so with that brief introduction of the overall our study design and our disease focus, I'll hand it over to my co-presenter, Amy Turner, who leads this project, and she will walk you through some of the experiments we're currently conducting and the technologies we're using, as well as the analysis approach we are utilizing right now for this data set.
Absolutely. Thank you very much for that great introduction to our research. And thank you everyone for listening to our presentation. We're really excited to share what we have here. So I'm going to go a little bit more now into our study design, looking in particular at the inter-individual differences between microRNAs, both intracellular and exosomal, and mRNA expression, and some of the relationships between those. For our first slide here, we just have the study design for this particular experiment. This study was designed to include 6 different individual cell lines. They were from 6 unique donors, all female, all African American, and 3 are unaffected. So the donors had normal left ventricular mass, and 3 are from moderate to severely affected LV mass representing LVH. What's critical here, though, is both gender and ethnicity of the donor cell line can substantially affect expression in the cell model. So matching both of those things is critical for experimental design. So the very first step that we did after generating the cell lines was to characterize these cells and look at, in our model, do we see mRNA expression differences that reflect the phenotype of the donors? So do these cells have differential expression for markers of hypertrophy? And when we looked at the results, we did see that yes, indeed, these cells do express classical hypertrophy markers differently between the affected and unaffected cell lines. As you can see here, this is the samples. 6 samples were represented here, 3 from donors with a high LV mass and 3 from normal LV mass control lines. The library preparation was done on the Illumina TruSeq, and sequencing was about 50 million paired end reads per sample. And here you can see we had significant differential expression in MPPB, MPPA, ACTA1, CORN, as well as other markers of hypertrophy. So after establishing that our cells are recapitulating aspects of our disease phenotype, the next step was to get a more in-depth, more comprehensive look at what was going on in our model. So for that, we moved our data into the Ingenuity Pathway Analysis, or IPA. And this allowed us to ask what's next and to do some more complex modeling, understanding some more of these RNA interactions that were happening. So for the mRNA expression analysis, the first step was to determine what we were loading into IPA. So for our analysis, we looked at genes with differential expression between the 3 LVH and 3 control cell lines that had a false discovery rate of less than or equal to 0.05. The raw data for this was aligned to HG19 human reference using RNA-STAR, and the gene recat was based on gene code version 19. The differential analysis was performed in R using DE-seq2, and we'll come back to that a little later when we talk about other ways to look at data to plug into IPA. And the 3 affected cell lines and the 3 unaffected samples were treated as biological replicates. So after the initial data was put together, it was uploaded into IPA, and there the first step was to create a core analysis. And the analysis type we're going to look at is expression analysis, and then the measurement type is the expression log ratio. So after running a core analysis, you get a summary screen, and on that summary screen, there'll be several metrics that will show different outputs and different summaries of your data. So for our analysis, we wanted to look at several things. The first thing we can look at is the analysis settings, which just give an overview of the different metrics that went into your analysis. The next parts that we can look at include top canonical pathways, which represents enriched pathways that are significantly differentially expressed between your different cell lines. So this was between our LVH high and LVH normal cells. Those were the enriched top canonical pathways that had differential expression. The next step was to look at top diseases and tox factors and biofactors, And from there, we can look then at upstream regulators, disease and biofunctions, tox functions, top regulators, top networks. All of these things are ones that were enriched in the data sets that were differentially expressed between our 2 cohorts. And here we can see again top disease functions. And then when we look at top tox functions, for ours, it was great to see that we saw cardiotoxicity was highly enriched. And then we can see the top tox list included cardiac hypertrophy, as well as other markers that fit in with the disease phenotype of the patient. Then if we look at top analysis ready molecules, this pulls out the top molecules that showed significant differential expression between the two cohorts. From here, we can move on to canonical pathways. So IPA allowed us to enrich for the top canonical pathways that were differentially expressed in our two data sets. Here we can go to then custom charts, and we're able to customize to look at whatever different metrics we want to filter our data by. And from there, we can move on to upstream analysis, and this allows us to look at activated or inhibited molecules in our pathways that are predicted to impact different networks. And from here, we can also customize the table. And what we can see here is that we can look at other metrics such as bias term, p-value of overlap, and we can select and filter this way depending on what we're interested in. The next step that we can look at for further narrowing down our disease phenotype is diseases and functions. And on diseases and functions, it will show which, either decreasing or increasing, which diseases and functions and which pathways are significantly enriched in our phenotype. We can see here this also enables you to select individual molecules that are found in the different pathways, and then to expand upon those molecules with the background information in the literature. The next step would be to look at regulator effects, and here it'll pull out regulator pathways and regulator molecules that are significantly expressed in our data sets. And here we can also then expand upon different diseases. So for this one in particular, this was cardiogenesis. So this was a pathway that is significantly differentially expressed in our data sets between the donors that had LVH and those that had normal LV mass. IPA also allows us to pull out the Tox-List Enrichment. And here we can see the bar graph showing the subset of the significantly enriched toxicity list using the differentially expressed data in our data set. And interestingly, heart failure, cardiac arrhythmia, cardiac dilation, and infarction, as well as several other known disease pathways are significantly enriched in our data. The next step then would be to see is to break down these tox lists and these things more so to look at networks. So what networks are significantly enriched in the data set? And the network tab allows us to pull up the most significantly enriched networks that we find differential expression in. And interestingly, cardiovascular disease was our top enriched network. So by selecting network 1, we're able to expand it further, and it will pull out all of the molecules and their interactions in this particular network. And then it will overlay our actual data, our actual expression levels that were differentially expressed between our two data sets, with green being lower in expression and red being increased in expression between the data sets. This can further be expanded by clicking on overlay, which will then allow us to overlay different diseases and different pathways with our data set. So for example, if we were interested in looking at how cardiovascular disease overlaps with this particular pathway, we would select that, perform it, and then it would highlight all of the molecules that in addition to this network were also present in cardiovascular disease. And then based off of the color of the circles, we could determine which molecules in our pathway overlapped with those with differential expression. The next step then would be to actually use IPA to look at pathway analysis with gene expression changes. So by selecting on the pathways, we can overlay our data with those pathways, again with the same scale showing increased measurement and decreased measurement, and see which aspects of specific pathways that our data is enriched for. This allows us viewing of a pathway with gene expression changes. In addition, the IPA also allows us to do pathway expression predictions. So this will overlay our actual data then with literature-based or knowledge-based findings. So is our data or our findings consistent with what the literature says these pathways should be doing? For example, if we see overexpression of a molecule, is that consistent with what literature says in this particular pathway should be happening in association with our phenotype? From there, it's capable of predicting the upstream and downstream pathway expression changes based off of the input data. And we can then further expand the pathways by selecting these networks, and we can grow them out and look at additional molecules that we saw in our data set and determine if they're also consistent or inconsistent with what literature is showing for these different pathways. So at that point, we've fully or more further characterized our mRNA expression differences, and we have seen that we do see significant differences in the donors, are the cell lines that come from donors with normal LV mass and those that come from donors with left ventricular hypertrophy. So then my next step, or my next interest, was to look at the microRNA expression between these cell lines. So the study design for our intracellular microRNA analysis was generating next-generation sequencing libraries prepared from 50 nanograms of total RNA using the QIAseq microRNA library kit for each of our 3 LV mass high and 3 control cell lines. Sequencing was performed on the NextSeq 500, and then sequencing output was mapped to miR-based version 21 using the QIAseq microRNA primary analysis pipeline. And this maps the raw reads to the microRNAs as well as clusters associated with the unique molecular indices and to count the unique microRNA molecules. And we'll talk a little bit about the UMIs in a minute. So the first step then is, I'm just going to first go through kind of a quick overview of the QIAseq microRNA workflow. The first step is to extract the sample. So this is RNA extraction. This can be done with several different kits. It can be the miRNAs kit, the ExoRNAs kit, the PAXgene microRNA kit, or really any other kit that generates high-quality total RNA. The next step is to then perform the cDNA synthesis and library construction, and in this particular case, it was done using the QIAseq microRNA library kit. Then sequencing is performed, and that's done on several next-generation platforms. It could be Illumina, Ion Torrent, or any other platform that's compatible with the library kit. And then data analysis is done. The first step is to use the QIAseq microRNA pathway, and we'll look at both primary and secondary analysis. And then the data interpretation is done using IPA to take the primary and secondary analysis and plug that data into IPA and really do an interpretation of our results. So just to kind of explain this a little bit further, the QIAseq microRNA library construction kit utilizes a really powerful technique when it ligates on a unique molecular indice. At this point, during library construction, this unique index is assigned to each individual molecule of RNA. And an example of the way that this can help with your sequence analysis is shown on this slide here, where you can see, for example, we have 2 genes. Gene 1 in the original RNA, whether this is from a single cell or this is from a pool of RNA, gene 1 has 3 transcripts, gene 2 has 2 transcripts. You integrate the UMIs, so every single unique microRNA molecule at this point is assigned a unique UMI tag, and then those go through the library prep and through sequencing. And here you can see that even though you started out with 3 to 2, a ratio of 3 to 2, after sequencing you end up with a ratio that is 2 to 1. However, if you go back and de-identify based off of the unique molecular indices tags, you can actually see that your true ratio is 3 to 2 based off of the number of unique molecules that were in your original data set. So this is a really powerful tool to help remove amplification bias or sequencing bias from your data. Another really interesting or really nice feature of the library prep is that the QIAseq microRNA kits are the libraries generated from these are significantly enriched for microRNA libraries and miss out on a lot of the adapter dimer that you can sometimes see when you're sequencing microRNAs. So after we did the QIAseq microRNA library prep, the first step then is to do the primary data analysis. Primary data analysis is done with adapter trimming, read mapping, and then UMI counting. If you go to the QIAGen.com GeneGlobe website, there actually are software pathways to do this, but some of the things you can look at here are the species. So you can look at human, rat, mouse, or other. In our case, it was human. You can do adapter trimming to remove the adapter sequences from the reads. You can do read mapping to identify the possible positions of the reads within several reference sequences. And then you can align the read sequences to your own reference sequences. Then you can also do UMI counting, and that will merge the unique UMIs with the mapped reads. So the next step after that is primary data analysis output. And what you'll get from that is a summary report, and you'll see a summary tab. You'll have a microRNA tab that has the mapped reads and the UMI counts for each of the microRNAs or other small RNAs found in miR-base. And then you also have a tab that's not characterized mappable reads, which will have mapped reads and UMI counts for each of the sequence that align to the genome. They'll look something like this depending on what your data input was. So at that point, you've done the primary data analysis, and then you move on to the secondary data analysis. And the secondary data analysis can also be done through the QIAGen site. And what you can do here is you can perform differential gene expression. And there are several ways to normalize the data. Some of these are listed here. We did DE-seq2 in our data set. And then what the output you get from this is, instead of just read counts, you now have fold regulation results, as well as scatter plots or some other ways of visualizing the data. and you have a file that's uploadable into IPA Analysis. And here is an example then. This is after we did all of the sequencing for the microRNAs, for the intracellular microRNAs between our cohorts. We looked at the, we aligned to UMIs, and then we looked at the differentially expressed genes, and the differentially expressed microRNAs. And then that file was uploaded into IPA and a core analysis was performed. And from here, we can then look at several different features. We can see not only the differential gene expression, but it also enables us to look at mRNA target prediction. So we're able to upload, since we had mRNA expression data for the same cells that we had the microRNA expression data for, we can upload our data set that has the differentially expressed mRNA in our cohort and align it with our differentially expressed microRNA. And then that will allow us to look then at target prediction. So what we want to do now, though, is we want to look at expression pairing. And the expression pairing will allow us then to filter our microRNAs based off of those that behave in the way that we would expect with an inverse relationship. So our interest, since we're looking for microRNAs that are potentially regulating differential gene expression between our two cohorts, is to look at microRNAs where the microRNA is up and the mRNA is down, or the microRNA is down and the mRNA is up. And this will allow us to filter for those. And at that point, then you can see that we've narrowed down our list of microRNAs to 277 microRNAs targeting 76 genes. Now, these are ones with differential expression in both the microRNA and the mRNA between our two data sets. The next step to further narrow this down to microRNAs of interest to us are then to filter by specific pathway. So this will enable us then to filter by pathways that are of interest. So for our disease phenotype, we're interested in cardiovascular pathways, so we would filter by cardiovascular signaling. And that further narrowed it down to 27 microRNAs targeting 10 mRNAs. We can then expand upon this and look at the different pathways that are affected by these mRNAs and microRNAs. And here you can see you can again do an overlay, and you can overlay specific disease and functions onto your particular microRNAs. So this allows us to see in between our two data sets, so in between our healthy cohort and our disease cohort, which microRNAs are differentially expressed, and also which mRNAs are differentially expressed, and then which pathways is this affecting. This allows us to expand upon the data further here too, where we can upload, again, additional data sets. So here we can see in our mRNA expression level, we actually can see the expression direction and which of our targets was affected in this particular cardiovascular pathway. And here we can continue expanding on this, and then this allows us to do the prediction also of our results consistent with literature for these particular pathways. So are we finding something that someone has shown to regulate cardiac hypertrophy and that we're also seeing differential expression for in our data set? Oops. So now we're jumping to the next topic. So at this point, we've characterized the mRNA and the intracellular microRNA in our data set, and we have shown that based off of the phenotype of the donor, you do see differential expression profiles and expression patterns between our cardiomyocyte cell lines. So the next step for us then was to look at, are we seeing differential expression between our phenotypic cell lines when we look at the exosomes? And a little bit of background on exosomes is that exosomes are packaged into, so they're microRNAs, immature microRNAs, mature microRNAs, and other RNAs that are packaged into vesicular bodies that are then released into the extracellular space. Here. And then this is just an example of what can be found in these vesicular bodies. It includes a whole host of different microRNAs from many different families, as well as small RNAs, proteins, and other elements that are released by the cells into circulation. So our interest then is looking at what role are these exosomes playing in the cardiovascular system. Since our disease phenotype is left ventricular hypertrophy and heart disease, we want to see what specific microRNAs or exosomal microRNAs are affecting our cell model, and what are they doing, and what are they? So we want to look at the exosomes in relationship to the cardiovascular system, and there is some knowledge now based. It's known that exosomes are released from cardiomyocytes as well as other cell types into circulation in the human body, and those exosomes are taken up, and the microRNAs are shared between different cell types. So what we wanted to look at then was, in our model of left ventricular hypertrophy and our cell line model, what are the differentially expressed microRNAs? So the first step to do that is to isolate the exosomes and prepare the library. So exosomal isolation and total RNA isolation was performed using the ExoRNeasy serum plasma kit from QIAGEN, which can do both serum and cell culture media. This is a really nice image of what you actually get from these. So ultracentrifugation is a well-established method of isolating exosomes from both serum and cell culture media. However, you do see kind of broken down unidentified structures or particulates that aren't quite matching the size, that look like they may be ruptured vesicles, whereas the elute from the ExoEasy kit, you actually see a higher ratio of intact vesicles with a higher purity. So the next step was, after we isolated the exosomes and isolated the RNA from those exosomes, library generation and sequencing was performed exactly the same as it was for the intracellular microRNA. So the RNAs were assigned the UMIs, they were run through the HiSeq, they were then analyzed doing the primary and secondary analysis, and then those results, the differentially expressed microRNAs from the exosomes, were plugged into IPA for additional pathway analysis. And here we can see that you upload it and do your core analysis, and it shows, like we saw with the intracellular microRNA, we actually see all of the differentially expressed exosomal microRNAs. And from here, we can actually then add in an mRNA data set of experimentally validated or predicted targets based off of several repositories of microRNA targeting, kind of primarily looking at predicted 3' UTR binding sites, some of which have been experimentally validated or just computationally validated. We can overlay that data with our data by uploading them, selecting the microRNA target filter. And then that enables us to show all of the predicted targets of our differentially expressed microRNAs and our exosomal fraction. And here again, we've narrowed down our list of microRNAs. We can also sort by what was the data set used to predict the targets. We can sort by what was the confidence. So are these mRNA targets ones that have been validated experimentally, where people have shown they are confirmed targets, or are these just ones with moderate or high experimental prediction? And then we can also look at then also filtering down by like pathway. So in our case, we would be interested in or potentially adding a path. So we could also look at disease or location or molecule type. So there are several ways to filter down your data to look for what you're most interested in your model. So our interest is looking at cardiovascular disease again. So at this point, we can narrow it down to 147 microRNAs targeting over 17,000 different genes that have been predicted using this. So if we do narrow it down to cardiovascular signaling, we've now narrowed it down to 147 microRNAs targeting 1,025 predicted targets. And then from here, we can expand upon the different pathways, and we can see then where our targets, again, lie in these pathways. And then further overlaying it. And so at this point, we've looked at the differentially expressed mRNA in our data set, the differentially expressed intracellular microRNAs, and the differentially expressed exosomal microRNAs. And here we can see in the differentially expressed exosomal microRNAs, we see pathways such as cardiac hypertrophy signaling, NOS signaling, eNOS signaling, and then the role of MFAT in cardiac hypertrophy. So then the next step then is to start thinking, how do the exosomal libraries, so the differentially expressed exosomal microRNAs, how do those compare to the differentially expressed intracellular microRNAs? And IPA allows us to perform a comparison. And with those, you would select compare, and then you would upload your intracellular and your exosomal data sets, or whatever two sets of microRNAs you were interested in looking at. And then you would select compare, and it will allow you then to compare the ratio of differentially expressed microRNAs that are unique to one set, unique to the second set, and then what microRNAs are differentially expressed in both data sets. And interestingly here, we find that distinct microRNAs are differentially expressed in the exosomal and intracellular fractions, and this is really pointing towards a more complex mechanism of microRNA packaging into the extracellular vesicles. And then if we further expand upon that idea, if we look at the predicted targets or the validated targets of our intracellular versus exosomal pathways, we can see that canonical pathways for the intracellular targets include things like the cardiac hypertrophy signaling and Wnt signaling. But if we look at the canonical pathways predicted for our extracellular microRNA gene targets, we start to include things like eNOS signaling and additional networks. And what that's really kind of showing here is that the overlap in pathways, such as MFAT or cardiac hypertrophy, in both the intracellular and the exosomal microRNAs likely points to a role in a more cardiomyocyte-specific kind of cell-cell neighbor cell communication, whereas the exosomal-specific targets like eNOS, NOS signaling, or even something like cardiac fibrosis may be for distal cell-cell communications, for example, cardiomyocyte to endothelial cells. So in summary of this, we hope that we've shown here is that we've established an in vitro model that comprehensively describes the role of microRNAs in cardiomyocyte hypertrophy. Our model has shown that the hiPSC lines from our LVH donors exhibit aberrant microRNA expression patterns both intracellularly and exosomally. We've demonstrated the complexity of the exosomal microRNA assembly by distinct intracellular and exosomal microRNA expression. And then our findings highlight that there's a really complex role these circulating microRNAs are playing in the multi-system development of cardiovascular disease. And in conclusion, I'd just like to thank everyone at our group at MCW, all of our NIH U01 participating groups, Jonathan at QIAGEN, and then just the NIH and our American Heart Association. And I'll turn it back over to Uli.
Well, and let me also finally say thank you to all the participants, and we also like to thank QIAGEN for their support and collaboration on this project. I hope you felt this presentation be helpful for your research and informative. And as said in the beginning, we will be very happy to answer any questions you have related to our presentation today.
Thank you, Dr. Broeckel and Ms. Turner, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window, type your question into the box that appears on your screen, and click the Send button. We'll answer as many questions as we have time for. Let's get started. Our first question is, what do you think are critical components of microRNA sequencing and analysis?
That is a very good question, and I think we in our presentation we already alluded to some of the things what we think is challenging. So first of all, and I wanted to emphasize this: if you work with cell culture and you're trying to analyze, as we do, different cell types or cell lines from different individuals, I mean, your overall cell culture and phenotyping approach really needs to be very robust. So we did spend a lot of time on testing for this, how reproducible our results are, to really optimizing our cell culture, cell plating, and cell maintenance, as well as RNA collection procedures. So if you're looking at different individuals, I mean, if you just have variation in the way how you culture and maintain and collect the material, that obviously would just bleed through, and you would detect it as differences between the different cell lines, when in fact it's really truly technical. So I think we didn't describe that in detail. This is something but we did spend a lot of time in optimizing this. And I would refer to Amy to emphasize some of the technological or the approaches around mRNA and sample collection and sample sequencing.
Thanks, Uli. I think that was a great explanation for that. And also in particular for microRNA sequencing and mRNA sequencing is that the quality of your input RNA is critical to the outcome of your experiment. If you have degraded RNA or low amount or contamination, that can significantly bias your results, as well as QCing your library prep after generation of your RNA-seq libraries. We kind of hinted at it in here, but with microRNA sequencing, it's very easy to have a lot of your reads eaten up by adapter dimer, or if you have degraded RNA, by just small degraded mRNAs that go in. So the quality of your input RNA is critical, and as well as the generation of your libraries. Those are two of the really critical components, pre-sequencing, that go into microRNA sequencing.
Thank you. Our next question is, how do you explain the difference between intracellular and exosomal microRNA?
That is also a very good question. So we, obviously, we maintain a single cell type culture. So in our cell culture, there are really only these iPS-derived cardiomyocytes, and the cell culture media doesn't contain any microRNAs, as it is serum-free. So the microRNAs we find in exosomes are truly from the cardiomyocytes in cell culture. Now, if you're again, we didn't describe this in greatest detail, but the sampling, when we sample the cell culture media for the exosomal analysis, this is about a cell culture media where the cells were maintained for 24 hours. And when we then for the microRNA, the intracellular mRNA and microRNA analysis, that's obviously at the end of these 24 hours. So to some respect, the exosomal fraction really reflects the shedding or the transport of release of microRNAs in the cell culture media over a time period of 24 hours. And obviously, it's quite perceivable that certain microRNAs turn on at different time points. So that will be one explanation, but the other explanation is, and I think there's also growing evidence that there are active processes which package microRNAs and other components which you can find in these vesicles or in the exosomes actively get transported because these cells actually, we see this as a mechanism to both interact and communicate with cell types close by in a distance. So those are probably the most likely explanations for these differences. But obviously, I mean, most of the microRNAs which we see, are also overlapping.
Thank you for that answer. Our next question is, what are your next steps to further explore your results?
I guess the next steps then to further explore our results are going to be to look at the communication between the exosomes in our cardiomyocytes into different cell models. So what are the functional impacts that these exosomes are having both on other cardiomyocytes as well as different cell types? And then perform expression analysis and functional assays of those as well.
Yeah, and in addition to that, I mean, as we're trying to understand the unique patterns, what we see in different individuals, and obviously, a more long-term or the more general goal of this is that we understand disease characteristic variation between different individuals. We understand that better and potentially can use this information to develop biomarkers and then also use this to develop potential new treatments. So, as we understand expression, as we understand the roles of microRNAs and exosomes, the big, really overarching question is how can we translate this to potentially diagnose patients better, more precisely, and with this information, how can we identify potential novel treatment options?
I would like to once again thank Dr. Broeckel and Ms. Turner for their presentation. I would also like to thank QIAGEN for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 10th of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.